Okay, so today we're going to have a look at enzymes, and in this video we're going to look at the structure of enzymes. We're going to look at two models of enzyme activity, which is the induced-fit model and the lock and key model. And then we're going to look at what these words mean. So you have intracellular, extracellular, anabolic and catabolic reactions. And more towards the second part of our lesson, we're going to look at different graphs to show how enzyme activity is affected by a range of factors, and we're going to describe trends and explain them based on these graphs. So what I'd like you to do first is think about what you can remember from GCSE biology. Think back to the different diagrams that you knew. Maybe the lock and key has stirred up a few words, and I want to see how many of the following key words you might know definitions for on A level. Here's our list of words. In the booklet that I've provided, you have a range of different reactions and different types of molecules that you need to define. So draw some lines, match them up, and then let's go through the answers together. So the first definition that we'll look at is anabolic reactions. These are reactions that build molecules. They're making reactions. They're the opposite of a catabolic reaction, which is a reaction that breaks down bonds between molecules and splits them apart. The metabolism contains both the anabolic and catabolic reactions. A catalyst is a substance that lowers the activation energy of a reaction, and we'll talk about this in a bit more detail soon. The metabolic pathway is the sequence of enzyme-controlled reactions. If you think of it like a factory production line, you have to process your materials a little bit, you have to make maybe the frames and cases and different parts of a product before you assemble it towards the end and have your finished product. So the metabolic pathway is a biological equivalent where you start off with one enzyme doing a reaction, catalyzing a reaction, and then you pass on that product to the next set of enzymes that will take the reactions a bit further until you finally have your finished product. Specificity describes the ability to only catalyze specific reactions, so only certain types. Your substrate is the molecules that the enzyme works on, and your product is the molecules produced by the enzymes. Let's look at the enzyme's shape next. So we know that enzymes are globular proteins and that they have active sites, which are shown in pink over here for the catalase enzyme, and this is sort of cut out space here on our little diagram. So we can see that the active site is dependent on the shape of the substrate because they need to have a complementary fit. And the way that enzymes work can be described using an activation energy graph first. So grab your booklets and let's annotate it together. We're going to start off with the initial energy state of your substrates. We've got this big hill looking thing, and then you have the energy level of your products towards the bottom. And what you notice with that initial increase over here is that you have to put in some energy in order to make the reaction happen, and then it will go and make the products. So when we don't have an enzyme present, the activation energy of an uncatalyzed reaction is going to be a lot higher. In other words, you have to put in more energy to get it going. This is different when we have an enzyme present. So the activation energy is significantly lowered, so much less energy needs to be expended in order to make the reaction occur. So another way that enzymes are going to reduce this activation energy is by forming something called an enzyme-substrate complex. So let's look at the different stages of forming this together. What we're saying is that the enzyme can do a number of things to make a reaction more likely to happen and lower its activation energy. And one of them is when the substrate fills the active site, when it interacts with the active site, you form this enzyme-substrate complex that might hold the molecule in a more favorable position to make or break bonds. In this case, you can see we've got our blue substrate, and then after the enzyme has catalyzed the reaction, we're left with two products that are left inside the active site of the enzyme, which are then going to dissociate and leave the active site so that the enzyme can be reused as a biological catalyst to process more reactions. Let's look at the difference between anabolic and catabolic reactions next. So in an anabolic reaction, your enzyme is going to bring your substrate molecules together and form a bond. Catabolic reactions are breaking, so you can see the bond starts off in the middle there. You already have a molecule that's joined together. and it's going to hold the molecule in a position that makes the substrate more easy to break the bonds between, so that two separate products will leave in this instance. Next thing that we need to look at is the difference between intracellular and extracellular enzymes. So what I'd like you to do is pause for a minute and briefly describe what's the difference between them. Intracellular enzymes, as per their name, work inside of the cell. So intra meaning within and cellular is referring to the cell. We'll look at some examples soon. Extracellular enzymes are ones that are secreted, so they're going to be pushed outside of the cell. And these are going to work in the external environment surrounding the cells. So that could be tissue fluid, it could be blood, it could be all sorts of things. So we have a nice table here. I would like you to use the internet to search around, and I want you to tell me what reaction is catalyzed, and I want you to tell me whether this would happen inside or outside of a cell, and whether it's an anabolic or a catabolic reaction. If you're confident, go right ahead. Otherwise, keep on listening to get a breakdown of what we're doing here. When we're filling in the reaction catalyzed part, that's fairly easy because you can have a quick search online and you'll notice that it's going to refer to a molecule minus the A's. So you should be getting results very similar to the way that things look here. When we're thinking about whether a reaction is intra or extracellular, you need to think about where you would find this material either within or outside a cell. So for example, if you're removing a carboxyl group in respiration, you need to think, does respiration happen within a cell or outside a cell? And that might help you guide this. The next is anabolic or catabolic. So as a reminder, anabolic reactions we know are to do with making bonds. Catabolic reactions are to do with breaking bonds. So in this example where we're removing a group, we need to think, are we making or breaking a bond here to decide what we should put in this box? When we talk about maltase, you should have researched what side of things are made from it. Do you find this molecule inside or outside your cells normally? DNA polymerase, where do you find DNA normally? If we're breaking down hydrogen peroxide, a byproduct of metabolism, where is metabolism occurring? So have a look through and give it your best shot. Let's go through our mark scheme then. So maltase catalyzes the breakdown of maltose to glucose in digestion. DNA polymerase joins nucleotides together in DNA replication, and pancreatic lipase breaks down triglycerides into glycerol and fatty acids. Let's have a look at decarboxylase and work out whether it's intracellular or extracellular. Well, as we know, respiration is a cell process that has to be within, because it's a removal of a carboxyl group, that means it's a catabolic reaction. Maltase again is breaking maltose. We know that digestion occurs in food. Food is outside the cells usually, so it's an extracellular reaction and it's catabolic because it's breaking. For DNA polymerase, where we're joining nucleotides together, DNA is found within a cell, so it's intracellular. We're making a molecule, so it's anabolic. When we're breaking down a byproduct of metabolism, that's a process within a cell, and it's breaking, so catabolic. Pancreatic lipase, again, is a digestion process, breaking down triglycerides into glycerol and fatty acids, and then that is breaking, so it's catabolic. The next thing that we need to have a look at is the difference between two popular models of enzyme action. We have the lock and key model that you might already be familiar with, and we also have the Induced-fit model. So pause the video here and please go and find a quick diagram and a definition of what happens in each and add it to your booklet, please. The first model of enzyme action is the lock and key hypothesis, and this assumes that the active site of an enzyme is rigid and it doesn't change in shape. So if you have a look at the way that the substrate enters the enzyme, they look like they're a perfect fit. So this tends not to be the case in reality. If you have a look at this animation here, you can see the structure of a protein and that it's very flexible and it's able to move and change. So crystallographic studies have indicated that proteins, rather than being fixed in place, as the lock and key hypothesis would indicate, are actually flexible. The induced-fit hypothesis is different, and it's very, very conditional on the presence of a substrate molecule in the active site. So the induced-fit hypothesis suggests that the active site is flexible and only assumes its catalytic conformation after the substrate molecules bind to the site. So we can see the substrate entering, and you notice that the active site changes shape in the presence of substrate to be a much better fit. When the product leaves, the enzyme is going to make its active site change shape again, because now we are not inducing a fit, so the active site reverts to its inactive state. Let's bring together a few lessons that we've done. So, based on what we've done so far and what we know about globular proteins from our last lesson, I would like you to take 5 minutes to explain how a change in the primary structure of a protein affects the shape of the active site and the function of an enzyme. So think of 5 different points. If you're confident, pause and go on ahead. Keep on listening if you would like some help. So for the first part of this 5 marker, you might want to focus on a few different things. We have the primary structure as one part of the question. We need to consider how that affects shape of the active site in the next one, and therefore the function of the enzyme. So we should be thinking of a few key words that were in our minds before. First thing that you should think of is, what does a primary structure look like? So when we're considering our primary structure, we are thinking of a single chain. Think of some keywords that you might use in order to explain how this chain appears and what sort of bonds are there. The next thing that we need to consider is how having this straight chain is going to affect its tertiary structure and its shape. So if we look at this molecule over here that has quite complicated shape, we want to have a little think about what sort of things might be holding that shape in place. What sort of parts of that protein are going to be forming the bonds that would hold them in place. If we change the shape of the active site, we need to think of what the consequences of that will be. So if you broke your active site, if you had changed your active site shape, what would have happened? Ultimately, because we're thinking about an enzyme function, think of what things interact with the enzyme in order to form products. So 5 marks. Make some notes for each of those 3 different bits you can split it into, and let's have a look at our mark scheme after. The first thing that we can talk about is that having a change in primary structure changes the sequence of amino acids, because a primary structure is just amino acids bonded together by peptide bonds. If you have changed the primary structure of amino acids, then you're going to affect the tertiary structure, because the bonds aren't going to be forming in the same positions between the R groups. So these bonds are ionic, hydrogen and disulfide. If the bonds aren't in the right places, then the shape isn't going to be the same either. So the shape of the enzyme and the active site could change. Because we've changed the active site, we know that the active site interacts with a substrate, so they will no longer be complementary. And if they are not complementary, we know that they can't then form the enzyme-substrate complex, where the substrate is inside the active site of the enzyme. Mark yourself out of 5 and please correct yourself to add on bits if you needed. The next part of our lesson is to think about summarizing what we know so far. So have a look at the sentences that I provided. If they're correct, just tick them off. But we're really interested in the incorrect ones. I would like you to go through and write the correct version of each of them, please. Looking through our mark scheme. The first part is enzymes are globular proteins. This is true. The second one is each enzyme has a specific active site that allows binding with any substrate. So this is conflicting here. If we're thinking about the part that conflicts, that is the any substrate. That is the direct opposite of what specific means. We could correct this to each enzyme has a specific active site that allows binding with a specific substrate. They do catalyze reactions that are intracellular and extracellular, but they do not increase the activation energy required for a chemical reaction to take place. In fact, they're going to decrease the activation energy. And for the next one, as they're biological catalysts, they do not get used up and they are not changed at the end of the reaction. Again, mark yourself out of 5 and correct the bits that you need to. So let's have a look at the effect of temperature on the rate of reaction. If we increase the temperature, we know that we're providing more kinetic energy to all the molecules present. That means there's more likely to be collisions between your enzyme and your substrate. And if they collide more frequently, you're more likely to form enzyme-substrate complexes, and that's going to increase your rate of reaction. If your temperature is too high, though, you're going to disrupt the bonds in your enzyme, and this will permanently cause a change in their shape. So if you imagine cooking something like an egg, you can't uncook it afterwards because you've broken those bonds, you've changed the protein structure, so it's permanently altered. If this happens in the active site of an enzyme, that means that you'll no longer be able to form a fit between your enzyme's active site and the substrate. So this will decrease the rate of reaction. We describe something as denatured when the enzyme's active site is no longer complementary to the substrate, so you cannot form enzyme-substrate complexes and the reaction cannot be catalyzed by that enzyme anymore. What I'd like you to do is describe the graph and explain what is happening here. So here you have the curve that describes temperature. If you're feeling confident, go on ahead and answer it away. Otherwise, keep on listening and we'll have a look at a few things you could talk about. In particular, you might want to mention these words. So if you would like a bit of help on this graph, my tip would be to split it into two different zones. We have a phase over here and we have a phase going downwards. Use these words to first off say what is happening, describe the trend between this point and this point. Afterwards, you need to go back and say, in this area here, this is happening because. From this peak downwards, this is happening because. Use these words to explain what's going on. So let's go through our answers together and state what's happening here. And I might be tempted to make reference to certain values just to help our explanation. So with our description, again, we're literally just saying the trend. So we've got between 0 and roughly 40 degrees Celsius, the rate of reaction increases. Afterwards, between 40 and let's say 52, 53 degrees, the rate of reaction decreases. To explain what's happening, let's make reference to the words on the side. So between 0 and 40 degrees, as kinetic energy increases with temperature, there are more collisions between enzyme and substrate molecules. This means it's more likely to form enzyme-substrate complexes, which increases the rate of reaction. From 40 to 53 degrees, the enzyme is starting to denature. This happens because the bonds break in the active site, changing its shape, leaving it no longer complementary to the substrate. This would be an example of something that you could say. So let's start putting this in your own words now. You have lysozyme as an enzyme found in tears. It can destroy some bacteria by breaking down the the polysaccharide chains that form part of their cell walls. We know that temperature affects the activity of lysozyme. The main part of your question is suggesting why increasing the temperature above 45 degrees Celsius causes a decrease in the activity. It's only 2 marks, you only need 2 points, but give it your best. Let's look through the mark scheme together. The first question, the first answer you could say is that this increased temperature changes the bonds in the enzyme. You could say bonds are broken. You can follow this up by saying the broken bonds change the shape of the active site, or you could say the active site is denatured. And we can round our answer off by describing the consequence. The substrate is no longer fitting into the active site, or the enzyme no longer catalyzes the reaction, or the enzyme no longer lowers the activation energy. You could also say no enzyme-substrate complexes can form. So score yourself out of 2. The next one that we can think there is about the primary structure of lysozyme, and it's a sequence of 129 amino acids. We know that the amino acids in positions 35 and 52 are in the primary structure, and we need to suggest how these two amino acids could be brought closer together to form part of the active site of the enzyme. So, if you're feeling confident, carry on, otherwise let's pause. Looking at what we've got so far, let's start drawing out some things that make sense to us. So we know that a primary structure looks a bit like this. We have a straight polypeptide chain filled with amino acids. We know there's 129 in total, so this would be position 129. We also have got positions 35, and we've got positions 52. We need to think about these being brought closer together, so I'm going to draw something. Let's say 35 is here, and here's 52, and they've been brought closer together. So what's happened in that protein molecule? We know that this is making part of the active site, but this is more a question about protein structures. So you need to think what could be happening between these two amino acids in these positions in order to hold them in that place that would then make them part of the active site. So you have 3 marks that we need to think about. Give it your best shot. Looking at our mark scheme, we can go through and have a look at a few of the main points. The first idea is knowing that if you change your primary structure into a more complicated shape, you're forming a secondary or a tertiary structure. You could give examples of a secondary structure by talking about an alpha helix or a beta sheet, or you could make reference to a tertiary structure by saying that you have a globular protein. You could say that the primary structure folds to also get a mark. After this, the idea of them being brought together is because the 2 R groups on the amino acids in that polypeptide chain are able to bond. And you could mention a few bonds and say that they're ionic, disulfide, or hydrogen in order to get your 3rd mark. I would like you to pause for a moment and think, why is peptide bond not accepted in this instance? So have a think. Our reason is because these bonds happen between R groups on different proteins. So if I had 2 peptide chains and I've got one R group here and I've got another R group on a different protein here, these can form a little bond in between. But peptide bonds are what is more in this backbone. In your primary structure, they are the bonds not between R groups, but between the amine end and the carboxyl end of a different amino acid. They're literally just there to join your beads on a chain, to join your amino acids together. So these bonds are not going to suddenly start forming between 2 amino acids in different places. So I'm going to pause here for this video, and in our next part we'll look at more factors affecting enzyme activity. If you have any questions, you feel welcome to email me. It's no bother at all. I'm more than happy to answer your questions, and if it still doesn't make sense after that, again, send some more emails. Yes, see you later.